Go.
Hello there! Today I'm going to talk about membranes. Membranes are barriers for ions and macromolecules such as protein and DNA. Membranes convey information and energy transformations occur across membranes. Membranes are composed of lipids and proteins. The first topic of this lecture is the energy requirements for membrane structure. In order to understand how membranes form and what they do, you have to understand water. So let me remind you about water. Water is complicated stuff. Water is the major component of cells. Water ionizes, it forms charged molecules. Water is polar, that is, it has an asymmetric distribution of charge. And water is capable of hydrogen bonding. You've seen this before. Water is about 70% by weight of cells. The rest is made up of ions, sugars, proteins, DNA, and all sorts of other good stuff. Water ionizes. H2O is in equilibrium with a proton, H+, and a hydroxide ion, OH-. The pH is the concentration of hydrogen ions, the negative log of the hydrogen ion concentration. So pH 7 is neutral, and that means that the concentration of hydrogen is 10 to the minus 7th molar in pure water. Water ionizes. Water is polar. It has a dipole moment. That's because oxygen is greedy for electrons and sucks the electrons in the orbitals towards it, away from hydrogens. Because the hydrogens, they don't really care so much about the electrons, not as much as oxygen does. So we have an unequal sharing of the electron bond, on the electron cloud in these chemical bonds. These are covalent bonds, but the protons are so weakly held that they can kind of come off. That's why water ionizes. You should remember that this structure is actually a tetrahedral sp3 hybrid orbital, and that two of these orbitals are occupied by hydrogen, but two are occupied by lone pairs of electrons. So it's a little tetrahedral with lone pairs of electrons sticking out from the oxygen. So because there's an asymmetric distribution of charge, that's called a dipole moment. And when you have salt in water, salt dissolves because the sodium ions and the chloride ions separate. Chloride is greedy for electrons. It takes the electron from sodium. The water dipoles orient towards the charges so that the dipole positive is facing the negative chloride, and the oxygen dipole negative, greedy for electrons, oxygen, is facing the sodium ions. So that's why it's a good solvent for salts. Water is capable of hydrogen bonding. So because this hydrogen is weakly held, it actually can partly bind to those lone pairs of electrons. And remember, those are in a tetrahedral orientation. These lone pairs of electrons will kind of partially attract, weakly bind hydrogen. So it's a weak bond, but it's there. And it all adds up to something. That's why water is liquid at room temperature, and that's why ice floats. because the liquid has a lot of hydrogen bonds, whereas the solid, some of those hydrogen bonds are not as well formed. So hydrogen bonds can occur between water molecules, and also anything that has an oxygen or a phosphate or an amine, anything that's charged. Any atom that has unpaired pairs of electrons, sulfhydryls, carboxyls, aldehyde, these have unpaired pairs of electrons that can hydrogen bond with the protons of water. So these are called hydrophilic. They like water. Hydrophilic. The opposite of hydrophilic is hydrophobic. Doesn't like water. Having aversion for water. So hydrophobic interactions are kind of weak force, actually. It's a tendency for nonpolar molecules to associate with one another in water. So these non-covalent interactions all add up to something. They're all weak bonds, but they are responsible for a lot of biological molecules. So these are hydrogen bonds, ionic bonds, that is charge separation. Van der Waals interactions, that is the electron clouds preferring to be at a certain proximity to one another. And hydrophobic interactions. So hydrophobic interactions are responsible for forming membranes. These weak interactions in general really provide the energy for a three-dimensional structure of protein and DNA, the interactions between protein and DNA, and the self-assembly of membranes. So that's the topic of today, membranes. Let's get back to membranes. Hydrophobic interactions can be thought of as causing water molecules to be more ordered. So this is a hard concept. Let me take you through it. In the presence of water, which can hydrogen bond, acetone dissolves. So acetone has this carboxyl group, which has those lone pairs of electrons, which can hydrogen bond with water. 2-methylpropane, this guy here, doesn't have anything to hydrogen bond with. It's nonpolar. The carbon and the hydrogens all have equal electron affinity, so there's no dipole moment. There's no separation of charge whatsoever. They all are pretty happy to share their electron clouds equally with each other. So there's absolutely no charge here at all. So water doesn't have anything to hang on to. And in fact, water becomes more ordered because it can hydrogen bond with itself, but it can't hydrogen bond with these nonpolar molecules. This is a key concept. If you remember our thermodynamics, free energy equals enthalpy minus the temperature times entropy. Entropy is randomness or disorder. So what happens when water is more ordered is that entropy is decreased. This makes the free energy more positive, which is energetically unfavorable. So hydrophobic interactions are really because water doesn't like to be more ordered. So these kind of avoid water and water avoids them. That's why we can have membranes. Fats are composed of, here's a glycerol molecule as a backbone. This is called a triglyceride. It has fatty acid chains linked to the glycerol through an acyl linkage. These are all hydrophobic. They don't prefer water, so they'll tend to associate with themselves. That's why we have membranes. Membranes are composed of lipids. Lipids have a polar head group and a hydrophobic fatty acid tail. Two of them typically. Why two? I'll tell you in a second. So this part is charged or polar. Doesn't have to be charged, could just be polar. It has molecules capable of hydrogen bonding, so it likes water. This part doesn't like water, prefers to associate with itself or with others of its kind, avoiding water. Lipids can be saturated or unsaturated. You'll see that in the ingredients of your food items. When it's completely saturated, it means there's no double bonds. When it's unsaturated, you have a double bond, and that causes a kink. You can't see my legs, but I just stuck my leg out. A kink in the lipid structure. And that makes the membrane more fluid when you have lipids with unsaturated, with double bonds in them. So lipids form membranes because they have two legs. Okay, so we have a glycerol molecule, two fatty acid chains. That's a kind of cylinder structure, kind of a cylinder. These cylinders prefer to associate with each other in a plane. If there was only one leg, it would form a kind of cone-shaped structure. And this would form a ball, or a micelle, preferably. It wouldn't form a plane. So just the structure of the lipid itself, when present with others of its kind, leads itself to form a membrane bilayer. And in 3 dimensions, the membrane bilayer prefers not to have any open edges, because all those fatty acid tails around the edge are ordering water too. So water doesn't like to be ordered. So because of entropy, the most stable structure is where the membrane bilayer is fully closed, fully sealed. And you can just make lipids and put them together, mix them up really well, and they'll form these things called liposomes. And they look like this. So the polar head groups are in red, the fatty acid tails are these little gray things here. So the exposed edges order water, decrease entropy, and therefore are unstable. So that's why we have membranes that form liposomes. So those are the energetic requirements for membrane formation. There are many different kinds of lipids, and they're distributed asymmetrically in the cell. So for example, in the cytoplasm, lipids that have negative charges prefer to be on the inner leaflet towards the cytosol, whereas the extracellular space, you don't find those. What you do find is lipids with carbohydrate or sugar molecules on them. It requires energy to flip these molecules, these lipids, across the membrane. And there are enzymes that do that. Flipase will allow, that's the enzyme that flips lipids. There's also one called scramblase that works in the endoplasmic reticulum during lipid synthesis. You can read about that. So enzymes are proteins, and there's a lot of proteins in the plasma membranes. In the membranes of cells, how do proteins span membranes? Let's think about that. Because the peptide bonds, here you have a nitrogen, you have an oxygen, all of these are going to be hydrogen bonding. They're all going to be hydrophilic. They'll like water. Here water is coming in. Water likes to orient towards those hydrophilic peptides. So how are you going to get this through the membrane? How is it going to be energetically feasible? The answer is that there are certain side chains on proteins which are hydrophobic. And in an alpha helix or a beta sheet, those side chains, which are hydrophobic, are facing out. So all these little R groups are the side chains. And if those are hydrophobic, those will prefer to associate with the lipids, fatty acid tails. An example of hydrophobic side chains is shown here. Alanine, which has a methyl group, valine, which has this structure, leucine, which is also just hydrocarbon, or isoleucine. These are all hydrocarbon side chains. And these dots here, these green balls here, are those hydrocarbon side chains facing out, which will quite happily be next to the fatty acid tails. They don't like water. They're hydrophobic. Similarly, in a beta barrel sheet, you have the side chains facing out, containing these hydrocarbons, associating with the fatty acid tails of the membrane. So there are lots of membrane proteins, and almost all of them, when they span the lipid bilayer, have either an alpha helix or a beta sheet. You can also have one side of an alpha helix be hydrophobic. You can also have lipid-linked or attached proteins, both cytoplasmic or peripheral. You can read about those in your textbook. Sugar groups, or carbohydrates, highly branched polymers of sugars, are attached to both proteins and lipids in the Golgi apparatus so that they're always on the outer leaflet. These form what's called a glycocalyx, that is an outer fuzzy edge of the cell. So they're on the outer edge of the plasma membrane. The attachment of sugars or carbohydrate is called glycosylation, and that happens in the Golgi apparatus. Membrane proteins perform a variety of important functions in the cell. They form the structure of the cell. There are enzymes that do various things. They help with the electron transport chain in mitochondria, which I'll tell you about later. They work for ion transport to conduct electricity in neurons. That's how we can think and study and dream. They transport molecules such as glucose for energy, and they're used to convey information. Ligands bind to receptors. Receptors are on the outside. They can carry a signal to the inside of the cell. So the cell is not all one kind of membranes. Their lipids, as I showed you, are distributed asymmetrically on the plasma membrane. Lipids are made in the endoplasmic reticulum, and they're transported throughout the cell by vesicular transport pathways. There, in addition, are specialized patches of membranes, which are like islands or rafts. They're called lipid rafts. And they contain lipids with longer, typically fully saturated, that is completely straight, fatty acid tails. So they're thicker than the normal plasma membrane. And they attract certain types of proteins which have longer membrane spanning domains. They have certain types of lipids that prefer and proteins attached to them. Lipid rafts are important for signal transduction and membrane traffic. They also contain cholesterol, which is intercalated, that is, wedged in between the fatty acid tails. Cholesterol is a lipid. It's used as a precursor to hormones such as cortisol, testosterone, estrogen, and those kinds of things. It's only got a little hydroxyl. There it is, a little hydroxyl. That's the entire polar head group. The rest of it is this hydrocarbon with rings. So this little hydroxyl sticks up with the polar head groups of the other lipids, and cholesterol intercalates, fits in between the other lipids. So lipid rafts prefer cholesterol, or you could say cholesterol prefers them. They have sphingolipids and selected membrane proteins. They also have the property that they're resistant to non-ionic detergents. So when a biochemist is making cell extracts, these will not be dissolved as easily in detergents. So membranes are really quite heterogeneous. And it used to be thought, in this model proposed by Singer and Nicholson in 1972, it used to be thought that membranes were a kind of two-dimensional fluid, and that proteins would diffuse quite easily. Well, that's kind of partly true. There are areas where proteins can diffuse and float in these two-dimensional membranes, but there are also areas like lipid rafts, where some proteins prefer to be. These are preferred zones. There are regions attached to the inner cytoskeleton, which form barriers, so those are barrier zones. And there are also proteins anchored to the cytoskeleton. Some proteins don't move at all. So it's a two-dimensional fluid, but it's really packed with protein. And some of the proteins are stuck, forming barriers, or they're stuck to the cytoskeleton. So finally, membranes are barriers. Because they have this polar region on the hydrophilic side, facing in and out, and they have a hydrophobic fatty acid tail, some molecules can get through and some can't. So let's consider what could cross a lipid bilayer. Here we have a synthetic lipid bilayer in a beaker, separating two sections of the beaker. So we have the polar head group, the fatty acid tails, polar head group, water on either side. What do you suppose can get through? Carbon monoxide, water, glucose, sodium, or chloride? The answer is that things that are really neutral, nonpolar, hydrophobic gases, such as oxygen, carbon monoxide, carbon dioxide, or solvents such as benzene or gasoline. If you spill gasoline on your hands, it goes right through your membranes because it likes those fatty acid hydrocarbons in the inner membrane. Small uncharged molecules, polar molecules, they don't get in very well. They'll get in a little bit, but not very much. Large polar molecules, such as glucose or sucrose, really don't get in very much, and ions are not transported. Because they're charged, the water molecules are holding on to them. And so you can measure the permeability coefficient of these. And it's 10 to the minus 12 in centimeters per second for charged ions. So they really don't like to give up their binding with water to get into those fatty acid tails. But water can go through a little bit. And these go through quite a lot. So, in sum, membranes are barriers to some things, to hydrophobic polar molecules. And these separate ions so that the cell has a different concentration of ions inside and outside. And it also has proteins inside that are not on the outside. Energy is transformed across membranes, and we'll talk about that when we talk about mitochondria and chloroplasts. And finally, information is conveyed across membranes. We'll talk about that when we talk about signal transduction and the electrochemical gradient. That's it. Thank you.